This is our video on cellular respiration, and we're going to start off with glycolysis. 2 molecules of ATP activate 1 molecule of glucose, which then adds 2 phosphate groups, which makes it fructose 1,6-biphosphate. It then splits into 2 3-carbon sugars, one of them being G3P and the other one being DHAP, although the DHAP can easily be converted into G3P. The G3P transfers electrons to the NAD+, which oxidizes the substrate and reduces the NAD+ to NADH. A phosphate group is then attached to the substrate. The substrate then converts into pyruvate, and this conversion produces 2 molecules of ATP. And this process happens twice. A carboxyl group is removed from the pyruvate, releasing a molecule of CO2. What is left of the pyruvate is oxidized, and the NAD+ forms NADH. Using its sulfur atom, coenzyme A attaches to the molecule. This forms acetyl-CoA. Now moving on to the Krebs cycle. The acetyl-CoA adds its 2 carbon acetyl groups to oxaloacetate, which produces citrate. 1 water molecule is removed and another one is added, which converts the citrate into isocitrate. The isocitrate is oxidized, so it loses a CO2 molecule and the NAD+ is reduced to NADH. Then the compound loses another CO2 molecule and is oxidized, which reduces NAD+ to NADH. Then an ATP molecule is produced by phosphorylation. The succinate that is produced oxidizes and 2 hydrogens are transferred to FAD, which produces FADH2. A water molecule is added, which rearranges the bonds in the fumarate and makes it malate. In the last step, oxaloacetate is regenerated by the oxidization and NAD+ is reduced to NADH. And then the cycle restarts. Now on to the electron transport chain. NADH donates hydrogen and electrons to complex 1 and FADH2 donates them to complex 2. The electron carriers take them to complex 3, then the cytochrome C, and then complex 4. Then they flow out of complex 4 to oxygen and hydrogen ions to form water. As this happens, protons are pumped from the mitochondrial matrix into the inner membrane space. The electric force and concentration gradient will cause the protons to flow through the ATP synthase enzyme. The enzyme also smashes phosphate and ADP to make ATP. Alcohol fermentation starts off with glycolysis, which results in pyruvate. A carboxyl group is removed from each pyruvate, which releases as carbon dioxide. The pyruvate is then converted into acetaldehyde. Then the acetaldehyde is reduced to ethanol by NADH. Lactic acid fermentation also starts off with glycolysis. Pyruvate is directly reduced by NADH and forms lactate, and NAD+ is regenerated without a release of carbon dioxide.